Now, so that's kind of stuff we did last time. So what we'll look at now is kind of the same stuff we'll do in lab. Sort of going to look at the anatomy, but we'll go into some details of little bits of it. So that's where we'll start. So here we're just looking, this is a diagram of the chest, thoracic cavity. Here's your diaphragm, lungs on your side. This is that part that's on your lab sheet. That's the mediastinum. It's one of those terms that put on your lab sheet. I'm not going to point to anything because it's too hard, but the mediastinum is just a column of tissue between the lungs, sternum, and backbone. It's where you find the heart. Of course, you find other stuff in there. You got thymus glands, you got trachea, esophagus in that mediastinum. It's that column of tissue in the middle. And so we're going to imagine you kind of pull out the heart, get rid of all this other junk. And the first thing we'll look at will be just the layers. So we did these in lab. Let's go into a little bit more detail. So here's a section through the heart, apex down here, base up here. If you start at the outside, say at the sternum, and you move in, we're going to cover all the layers. So what you have, you imagine this is the heart itself. It looks more like a prostate gland. There are membranes, again, associated, serous membranes, associated with all of your body cavities. The one associated with the heart, these are specifically called pericardial membranes. So what you find is there is a loose fitting membrane out here. It's just like a loose bag. Saw it a bit in the cat. It folds back on itself. And there's another layer that physically attaches to the outside of the heart. These are labeled. The outer layer is the parietal layer. And that's going to be true in all the different cavities we look at. Specifically in the heart, this outer loose bag is a parietal pericardium, where it folds back on itself and physically covers the heart, so it's in physical contact with the heart. It's a layer of the heart. That is the visceral pericardium. Most of your internal organs, a few exceptions, most of them have this double layer of membranes, and these are collectively called serous membranes. The space in the middle, that cavity, it's exaggerated on light membrane, but that's filled with serous fluid. This double layer of membrane secretes a lubricating fluid that allows the heart to slide around without a lot of friction because it's constantly moving. So that's the pericardial cavity, and that would be filled with serous fluid. Now, these serous membranes, again, you have them in the abdominal cavity. They're called the peritoneal membranes in the abdominal cavity. Around the lungs, you have the same setup. They're called pleural membranes. Around the heart, pericardial membranes. Those three sets of serous membranes all have one function in common. They all secrete that lubricating fluid so your internal organs can slide around. So that's true of the pericardium as well. However, they pick up some additional functions in the heart that you don't find in the serous membranes in the other body cavities. And you can tell this just because of their structure. Most serous membranes are delicate. They're basically a layer of epithelia and a basement membrane, and that's it. So if you, for example, go in the abdominal cavity, pull on the serous membranes, they'll just shred. They're very delicate. The heart, however, the parietal pericardium, this outer layer, has an extra layer of tissue on it that you don't find in the other serous membranes. And that's kind of weird. You find something, hey, it's in anywhere else, what's that there for? You look at this enlargement, it's what we're showing you here. This is the visceral pericardium here. It folds out, and this is a parietal pericardium here. But the difference here is the parietal pericardium has two layers. This is on your lab sheet. I typically don't ask it lab. What you have, just looking at the parietal pericardium, kind of this brown layer here, that's the serous layer. That's a layer of cells in the basement membrane that all the other serous layers in various parts of your body have. But what's unique here for the parietal pericardium, this layer out here, what they're calling the fibrous layer, that's a dense connective tissue. The same kind of junk you find, in tendons and ligaments and muscle fascia. It's very strong. This is a little weird because normally you don't find this fibrous layer on any other serous membranes. It's only on the heart. So why is it there? What do you have this fibrous layer for? Again, you remember the cat, when I dissect that, you can actually lift the cat up by the parietal pericardium. It's that strong, it's really tough. Why is it there, you think? I'm about to go off on a tangent here and tell you why it's there. It is protective. It's got nothing to do with the lubricating function. What I have to do to explain its function is I have to review or go over some material that I typically cover in the reproductive systems, but I haven't. You might have looked at it a little bit last semester. What I'm about to scribble on the board is something called a length tension curve or length tension diagram. This is associated with muscle. Now, we have looked at one type of muscle contraction already last semester. You looked at skeletal muscle contraction, right? Remember the sarcomeres and the myosin filaments and the actin filaments, right? That rings a bell a little bit. All right, let me look at skeletal muscle first, because you have a little experience with it. What I'm going to do, let me dab out this overhead for a moment. I'm going to set up a little apparatus. We have these up in the lab. We don't use them in the lab very much anymore, but this is just like a ring stand, one of those middle stands you put on your lab bench. So anchored to the floor here. There's a little threaded piece here, so there's a little threaded rod here, so you can raise this up and down. That's all it's supposed to be. This box here represents something called a muscle transducer. All this does is got a little hook hanging out of it. If you pull on that hook, this thing will send an electrical signal to your computer or oscilloscope, whatever you're recording. And it'll basically turn a mechanical force into an electrical signal. That's really all the muscle transducer is. It's an electronic device, like a mechanical device. If you pull on this hook, it'll give you traces. So for example, if you were hooked this up and you started recording, when you pull on the hook, it would go up and you let go and go back down. So it's just going to record tension. That's all this thing does. It's not really that complicated. Now what we're going to do is I'm going to take like a test tube here, fill it with saline, anchor it to the floor. I'm going to hook a muscle up to this thing. So I'm going to take a piece of skeletal muscle, imagine a piece of the biceps or something. I'll attach it here, run it down with this hook. So here's a piece of skeletal muscle. Now what I'm going to do, I'm going to hook this thing really loosely between those two hooks. I mean, it's not being pulled on at all. It's just got hooked in here, hooked in here. I'm going to put a ruler up here. I'm going to measure how long it is. I'll just stuck a ruler up this muscle, this long. On this diagram, this x-axis here will be the length of the muscle that you measure with that ruler. Then I'll just get a battery or something and I'll hook a couple of electrodes up here to a battery and a switch. So of course, if you electrically stimulate a muscle, it'll contract, right? That's all I'm doing. So I have this thing as short as possible. I stimulate it and the muscle will contract. Now when it contracts, it pulls down this hook a little bit and I will get a measure of tension. Say it's right about here. So this y-axis is basically the force or tension, force. It's how hard the muscle pulls down. So I get a little point, say, down here. Now what I'm going to do is I keep everything where it is, but I take this threaded part and I raise the hook up a little bit. And of course, as I raise it up, I'm going to physically stretch that muscle out. They're a little stretchy. Now the muscle is going to be stretched a little bit longer. So over here somewhere, I stimulate it again, and I record the tension. And I'll just keep repeating that. Take this thing, line it up a little higher. And of course, I'll stretch the muscle out longer, stimulate it again. I'll get some force here. And I'll just keep doing that to the limits of the muscle and setup. And what I'll get is a curve that looks something like this. Connect the dots, I'll get a line, kind of something like this. That is the length tension curve for a skeletal muscle. Now what you'll notice, depending on the length of the muscle, that's going to affect the amount of tension it develops, right? It's all the same. Now, why does this happen? All right, let me look at the extremes. Over here, what this means is there's basically no tension. Over here, basically no tension. Remember, these skeletal muscles are made of sarcomeres. So what you're seeing at this end, when you really stretch out a muscle, you're going to see something like this. That's the Z line. Remember that little disc of protein at either end. Then you have your actin filaments sticking out like this. And your myosin filaments between. All right, here's what happens. You remember the way you get tension in the skeletal muscle. You get those little cross bridges forming between the myosin and the actin, and then they'll kind of rotate a little bit. You're doing that sort of thing with all these myosin heads. The myosin heads, those cross bridges are called, they occur where the myosin and actin filament overlap, right? Imagine now what's happened here. If I pull this sarcomere enough, how much overlap do I have between the actin and the myosin? Myosin here, actin here, no overlap? Will you get any contraction? No, you've overstretched it. If you pull a sarcomere out enough so that the actin and myosin filaments don't overlap, you get no cross bridges, no myosin heads bind to actin, you got zero tension. That's what you have here. At this end, where the muscle's really short, you have something that might look like this, at least diagrammatically. Imagine sort of if your sarcomere gets really, really short, eventually the myosin filaments will smash into the Z line. They'll crumple into that disc. The actin filaments will get close enough together, they start crumpling up. That's what metal spaghetti lines mean. So when the sarcomere gets too short, the proteins start to it's protein interference is what they call it. They start to kind of crumple at either end. Kind of makes sense? Again, you get enough of that crumpling, you can't get any more tension. That's right here, no tension. So look smack dab in the middle, kind of somewhere in here, and what you'll find is, at this point, you have the maximum amount of overlap, but you haven't got any protein interference yet. That's where you get the most tension. Kind of makes sense, just from what you remember of sarcomeres. This is true of all striated muscle, like skeletal muscle. If you change the length of the muscle by stretching it, you'll change the amount of tension. Typically, the way your skeletal muscles are hooked up, they're almost at the optimal length, somewhere in here to start with, so you get maximum tension. Now, that's skeletal muscle. We don't care about skeletal muscle. But let me mention just briefly the other kinds of muscle, because we never look at anything but skeletal muscle in Anatomy 1. There's no time. Well, you do know about smooth muscle, right? Look at it in lab. And usually where I introduce this is when we're looking at the uterus, because here's the example I usually use. All right, when you have junior developing in the uterus, right? Now, most of the uterus is smooth muscle. What do you know about smooth muscle? Can you consciously control it? No, smooth muscle is involuntary, right? So here's the problem I have, at least for me, being a male that's had children. All right, you got that uterus made of smooth muscle, right? Junior little embryo implants in there, and junior goes bigger and bigger and bigger. And then some random time later, that smooth muscle starts to contract. Your wife grabs by the arm, honey, we gotta drive the luteal down. You drive 80 miles an hour, and it's terrible. Because it's smooth muscle, you don't have voluntary control over it. So my question is, why isn't the uterus skeletal muscle? That's under voluntary control. Think how much easier it would be. You wait about 9 months, and you call a doctor, hey, honey, I think Tuesday for an appointment. Yeah, and you call the doctor, make your appointment for Tuesday at 2. And Tuesday morning, you leisurely drive to Louisville, you might stop and get lunch, and then you go to the doctor's office. The doctor says, are you ready? You go, sure. You go, okay. Well, it's about that time. Okay, contract. Boom, out it comes. It'd be so easy, so much easier. There would be children born in snowstorms in the back of taxis. Why isn't the uterus skeletal muscle under voluntary control? It would be way easier, isn't it? Why isn't it? All right, there's a couple of reasons. How can we engineer it? Let's genetically engineer a uterus made out of skeletal muscle, so it's voluntary. It would be, again, so much easier. For everybody, scheduling works. Why not? Because here's the problem. If you made a uterus out of skeletal muscle, that sucker starts out about the size of your fist, right? But then junior grows in there and stretches out more and more and more. And after about 9 months, you're over here. That sucker stretches out tremendously. So 9 months later, you send a signal to the skeletal muscle, and oops, it can contract. So you'd be pregnant the rest of your life, which would probably be really uncomfortable. Skeletal muscle won't work because you can't stretch it that much. So smooth muscle, if I did exactly the same experiment, but put a piece of smooth muscle on here, and again, let it get really short, stimulate it, measure the tension, stretch it out, stimulate it, measure the tension, stretch, you know what would happen? If I did the same thing for smooth muscle, smooth muscle does not have sarcomers. It's not striding. That's why they call it smooth. One of the advantages of smooth muscle, why you find it in most of your internal organs, it is not sensitive to stretch. You can take a piece of smooth muscle, stretch the crud out of it, when you stimulate it, it still generates about the same amount of tension. That's why it's in most of your hollow internal organs. The uterus, your bladder, your intestines, almost everything, gallbladder, it's all smooth muscle because you can stretch it, and when you send a signal to it, it'll still contract with the same amount of force. And that's why. We don't talk about that much in Anatomy 1, but mechanically it's different. Now, what we're talking about is cardiac muscle. Where does that fall in this spectrum? Cardiac muscle is striated, all right? It is very similar mechanically to skeletal muscle. However, just keep in mind, when we're looking at length and tension and stuff like that, this is just real diagrammatic representation of the heart, but they're trying to show you the muscle fibers. Of course, skeletal muscles are very linear, right? They're attached here and here. In cardiac muscle, the fibers are wrapped around that hollow chamber. So when we talk about length in the heart, what are we really talking about? Imagine this is your ventricle. These are the muscle fibers. Stretch it out, make it smaller. Stretch it out. Length is related to circumference, right? Or probably the best way we can be to think about it, that hollow space inside, that's where the blood is, right? If I make that space bigger, I can fill it more, right? Think of stretch or length in cardiac muscle cells basically as filling. If you cram more blood into the heart, you're going to stretch it out, right? That's going to increase the length of the muscle fibers. So that's kind of the relationship. But if you did that, if you hooked up a heart muscle, just like a skeletal muscle, and it did a length tension curve, what you find is something like this. Now, assuming we're looking at an intact heart, it would be very similar to skeletal muscle. Instead of length, though, we might talk about filling or circumference. That's the relationship, right? Circumference in cardiac muscle is the same as length in skeletal muscles. That makes sense. So what we notice is, as you filled it more, you get more tension. Fill it more tension. Fill it more tension. So it's got the same relationship as skeletal muscle, except you'll notice my diagram stops right here. This is important. This is going back to that parietal pericardium. What's its function? Why does it have that fibrous layer? Remember, most of your hollow organs are smooth muscle, because if you stretch them really far, it won't affect your tension. Cardiac muscle is not. The way to think about it is you have this not very stretchy, sort of rigid sac around the heart. So the heart expands, where you stretch it more and more. Eventually what happens? It hits the parietal pericardium. This is its function. The reason in an intact heart with the parietal pericardium, you won't get past this point, because this is the stretch limit for an intact heart. That's what the parietal pericardium does. It'll allow the heart to expand only to a point, then it hits that sort of rigid bag and it can't expand anymore. The reason that's important, because what would happen if it weren't there, you'd overfill the heart. If you overfill the heart, just like a uterus made out of skeletal muscle, if you put too much blood into it, it couldn't contract anymore. Basically keel over dead. So that's what the parietal pericardium does. It's to prevent overfilling the heart, which would basically allow it to potentially get this downstream end of the length tension curve, which would be leaking. This concept's going to come up. It has a lot of impacts on the heart. Just this length tension curve, and again, the parietal pericardium, in fact, it has to stop there. I will mention, because this is where that article comes in, you don't necessarily need to read this article, pericardectomy in dog. It's you can first replace this paragraph long. But what they did in that experiment, it demonstrates part of the effects of this parietal pericardium. They took a bunch of their greyhounds, and they took a handful of them, and they did open heart surgery. They removed the parietal pericardium. Then they took another group, they called it the control. They do what's called a sham operation in physiology. They open the chest, but they don't do anything, and they just close them back up. It's just to see if the surgery itself had any effect. And what they were doing, more or less, was looking at cardiac output. A mammal's aerobic capacity is basically limited by how much blood and therefore oxygen you can get to the tissue. So what they did in these dogs, they basically monitored oxygen delivery, maximum oxygen delivery to the tissues. That's an indication of cardiac output. And after the two sets of dogs recovered, they would then race them and monitor their oxygen delivery to the tissue. What they found is the dogs, when they removed the private pericardium, had a greater ability to deliver oxygen to the tissues. They had a higher cardiac output. Now, it's a short-term study. They don't tell you what happened 3 years later. But the reason for that is the private pericardium stops the heart from filling a little bit before the peak. So if you remove this, what can happen is you can actually get some greater stretch and greater force of contraction. So it actually limits cardiac output. And the reason I mention this is if you look at cardiac output, of course, that's the amount of blood the heart pumps per minute. 5 liters per minute is what most people pump at rest. That supplies your baseline oxygen needs. But if you exercise, humans have the ability to pump about 8 times more blood than you're pumping right now. That is, an athlete can pump maybe 40 liters per minute, 8-fold increase. That's an athlete. Couch potatoes cannot. What happens when you want to increase cardiac output? Remember, cardiac output is determined by heart rate times stroke volume, right? Heart rate is determined by your nervous system. You could be a big old couch potato, and if you start exercising, your heart rate goes up. It's very easy to elevate heart rate. Stroke volume will also go up a bit, even in a couch potato. I think you can rise it by about 50%. But the reason an athlete can pump a whole lot more blood than a non-athlete, part of the aspect of aerobic training, working out hard all the time, it's this part, stroke volume. What happens in an athlete, the heart, when you exercise, well, not only does it get a little bit bigger, you see changes in the private pericardium. An athlete actually has a bigger private pericardium. It allows the heart to fill up. Their stroke volume goes up tremendously. That's why, any cross-country runners in here? No. Yeah? What's your resting heart rate?
I don't know right now.
No, I've had guys and gals that come in and do cross-country. Their resting heart rate is like 55. That's actually sort of abnormal. It's not for an athlete. That's lower than it should be. It's actually considered bradycardia. So if you have, let's say, an athlete, their heart rate equals, let's say, 55 beats per minute. And then you look at a couch potato, and my resting heart rate is 80 per minute. If we're both at rest, who's pumping more blood? The answer is they're pumping the same amount. It doesn't matter if you're an athlete or a couch potato. When you're sitting there doing nothing, your basic metabolic needs are about the same. How can that be true though? I'm pumping 80 times per minute and you're only pumping 55. Should I be pumping more blood than them? No. My stroke volume is smaller. So in order to keep an adequate amount of blood moving, I have to have a higher rate. The athletes who have a bigger heart, bigger private pericardium, their resting heart rate is lower because their stroke volume is bigger. That changes a lot with exercise, aerobic training. So that's kind of the function of the private pericardium. It limits stroke volume. And it can be changed with aerobic training. But that's the purpose of that fibrous layer. Basically, it prevents the heart from overexpanding. Now, one other thing while we're looking at this length tension curve, I got to mention because it's important for the heart. I'll mention here a few times. It is called Starling's Law of the Heart. It's not named after that black bird that floats around the yard. I think Starling was a physiologist. Anyway, this is really important. Now, your book will give you the formal definition for Starling's Law of the Heart. It'll say something like, the greater the length of the myocardial fibers, the greater the force of contraction. What does that mean? It's that latent tension curve. Here's my easy way to remember Starling's Law of the Heart. Basically what Storm's Law of the Heart says, if you put more blood into the heart, think about it, here's your heart, right? It's got these fibers wrapped around it. If I squeeze more blood in there, I'm gonna stretch it a little bit more, right? So when the heart contracts, guess what? It contracts with more force. Starling's Law of the Heart, basically what it's saying is if more blood flows into the heart, when the heart contracts, it's going to eject more. It will contract with more force. It's because of that length tension. Now, let me scribble back down here. Remember that length tension curve looks something like this for the heart. Stop there to the pericardium. This is filling or input. So if I fill up the heart with a little bit more blood, when it contracts, it's going to contract with more force. If I fill it up even more, when it contracts, it'll contract with more force, more and more. Does that kind of make sense? This is actually one of the important automatic control mechanisms of the heart. Without doing anything with your nervous system, thinking about it, nothing. If more blood flows into your heart, when it contracts, it's going to eject more. If less goes into your heart, when it contracts, it ejects less. It's an automatic control mechanism. There are several things that cause that blood more to flow in or less flow in. But that's what Starling's Law of the Heart. It's this length tension relationship. So if, for whatever reason, more blood flows in, the heart's going to pump more. And this is kind of cool because we draw a quick circulatory system over here. Maybe we're just at the pericardium. If you want to be really simple, here's your circulatory system. You got the heart, it's going to pump it out some tubes called arteries. It's going to pump to the tissues, and then go back to the heart. We have a closed circulatory system, right? Blood to the blood vessels of the heart only. But here's what's kind of cool. This area here represents the tissues that need the oxygen. And it turns out the blood vessels going into the tissues, there's like a valve there. As we'll see, it's just contraction of smooth muscle. But if these tissues are metabolically active, do you want them to get more oxygen? The answer is yes, right? That's the idea. If you want to run or exercise, you better get more oxygen to those tissues, right? Nod your head. Makes sense? When this tissue becomes active, what happens is this blood vessel will actually open up, almost like a valve opening. When this opens up, more blood will go to that tissues. But here's what's kind of cool. If more blood flows to the tissues, that also means more blood is going to flow into the heart. Because of Starling's Law, guess what's going to happen? More blood flows into the heart, more blood flows out. This is automatic. The actual sort of pinch point or control point is the actual tissues. If tissues are metabolically active, they'll sort of open up more blood flows through them, which is great, they get more oxygen. But also as more blood flows through the tissues, more blood flows back to the heart will automatically pump more blood. On demand. This is not required nervous system, nothing. It's an intrinsic sort of mechanical characteristic of the heart. So more blood flows through the system of the heart, it will automatically pump more. Less flows through the system because you need less, the heart will pump less. That's kind of that's what stroke body does. That's what Starling's Law of the Heart means. And that will pop up in several areas. We'll talk about this again. So we're done with the pericardium. We got the first layer. So the parietal pericardium has that fibrous layer on the outside, and it's sort of protected. It prevents the heart from overfilling and actually pumping with less tension. So it is important. Then, of course, it has the serous layer underneath. That's just for lubrication. Then you have the pericardial cavity. That's just filled with serous fluid. That's the lubricant. Then we get to the wall of the heart itself. And this isn't anything different from that. The outer layer of the heart is the visceral pericardium. But here you'll see it has two names. The outer layer of the heart, physical part, once you cut that parietal pericardium off, you're left with one bunch of fused layers. It is the visceral pericardium, but it's also called the epicardium. It's exactly the same thing. I don't care what you call it. That layer that's physically attached, again, it's just a layer of cells in the basement membrane. That is the visceral pericardium. It's also called the epicardium. Generally, by convention, if you're talking about the serous membranes, you call it the visceral pericardium. If you're talking about the layers of the heart, you call it epicardium. But it's exactly the same structure. I don't care which one you call it, for example, in lab. It's the same piece. And that's just a single cell layer. Now, most of the heart's volume, if you remember those hearts we sectioned in lab, most of the heart's volume, the thickest layer, is the myocardium. Just means cardiac muscle cells. Myocardium is the muscle layer. This is the layer that actually contracts, generates the force. You'll see the cells referred to as myocardiocytes. They are just cardiac muscle cells. That's the bulk of the heart. Most of its volume, that does all the work. Now, I need to mention a few things about cardiac muscle cells because you really haven't looked at them much. You looked at them in lab last semester where, you have to tell the difference between skeletal, cardiac, and smooth, but you probably didn't talk about it in the lecture at all. So let me take a minute to look at cardiac muscle, and we'll compare it to skeletal muscle, since in theory, since we've already got over that a little bit in anatomy one. Now, do you remember any differences between cardiac and skeletal? Just looking at the, for the lab, I know you can tell the difference. What was one of the differences?
Cardiac muscle striated.
Okay.
Also.
Yep. They are both striated. That is, again, they have the sarcomeres, active filaments, myocytes. The contractile mechanism is identical. I mean, the functional unit of contraction is a sarcomere for both, no difference there. That is how the same. If you remember anything else, how'd you tell them apart in lab?
Was the internuclear disc?
Intercalated disc. All right, now there's a lot going on in intercalated disc. I don't want to talk about most of it, but basically what you have where cardiac muscle cells join. It's basically a cell-to-cell junction. In cardiac muscle cells, if you have a good preparation that was stained properly, there's a dark line right where two cells butt up against each other. This is the intercalated disc. This is basically, in the histology slides, this is how you tell them apart. Cardiac muscle cells have that intercalated disc. You won't find that on any of the muscles. So it's a good slide, and you can see the intercalated disc. That's a giveaway. What are in the intercalated disc, though, is what's important for us. At this cell-to-cell junction, we're calling it intercalated disc, there's a number of different proteins. Some of them I'll just mention briefly. There are desmosomes in here. Anatomy 1 was so quick, I barely even know if I talk about desmosomes, but these are proteins that knit both cells together. So, of course, when these two cells contract, you don't want them pulling apart, right? That would kind of be disastrous. So, desmosomes, there's several different types, but think of them as a big clump of protein in each cell with all these fibers knitting them together and attached to the cytoskeleton. So there's belt and spot desmosomes. They're just a mechanical connection. Again, the reason the intercalated disc stains is there's a collection of proteins there. That's how you can stain. Those aren't going to be, beyond the mechanical function. I'm not worried about those. What you'll also find, the one that's important to us functionally, in the intercalated discs, there are things called gap junctions. Remember what those are? What's a gap junction? Anyone know? Again, I'm so fast and then I'm in 1 8 weeks, I don't remember, go hard memory. Okay, all it is, it's basically it's an electrical synapse. I know I talked about those in anatomy 1, but I don't know about gap junctions specifically. It is a protein, you can think of it as rolling into a tube so that the cytoplasm here is actually connected to the cytoplasm of the next cell. In the heart, every cell is connected by these gap junctions. Now, what they'll tell you about gap junctions, they're for cell-to-cell communication. Basically in the heart, because all the cells are connected with these gap junctions, what you're really looking at, the heart acts like one giant cell. I think the textbook will refer to it as a functional syncytium, which means it's acting like one giant cell. Because all the cytoplasm is connected. The reason this is important sort of functionally, think of skeletal muscle. Did you get to look at motor units in anatomy 1? I know I didn't have time, but in skeletal muscle, if you go in and stimulate one skeletal muscle cell, what's going to happen? That one skeletal muscle cell will contract, right? What will the rest of the cells do? Absolutely nothing. That's how you get creative contractions in your arm. You can contract a few cells for some tension, more cells, more tension, until you recruit the whole thing. Skeletal muscle cells are electrically independent. Cardiac muscle cells are not. In fact, you don't want it to be. Imagine if some of the cells in the heart contracted and some weren't, and others contracted, that would just kind of slosh the blood around like squeezing on two ends of a water balloon, right? That's not what we wanted to do. You want all the cells in the heart to contract at about the same time to squeeze on the blood and effectively, eject it out into the arteries, right? So cardiac muscle cells are acting like one unit because these gap junctions. That's why a pacemaker works. You know what a pacemaker is, more or less? In its simplest form, it's a battery with two wires and some kind of electronic circuit that sends out an electrical pulse 70 times a minute. Well, with a heart, you just stick those electrodes in one cell. When you stimulate that one cell, because of the gap junction, the impulse spreads to every other cell in the heart and it all contracts. If you put a pacemaker in a skeletal muscle, just that one cell would go twitch, 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 and wouldn't be conducted. So cardiac muscle cells are all connected with these gap junctions. That allows it to work as a unit, which is what you want. You need all the cells to contract at the same time to effectively squeeze on the blood and inject it out the heart. So skeletal muscles do not have those. All right, what else do you know that's different between skeletal muscle, cardiac muscle? I'll mention a couple just histology ones. They're funky, not that important. Cardiac muscle cells do tend to branch more in microscope slides. They're more branchy. Cardiac muscle cells are. Skeletal muscle cells are very linear. They're just big, long cylinders. That's it. So that's one way to tell looking at a microscope slide. I'll list it up here. The function is not that important to us. Also, the number of nuclei are different. You might have noticed in skeletal muscle cells, by the way, are huge. You can measure them with a ruler. You can measure them in inches, good fraction of a millimeter thick. But how many nuclei are in a skeletal muscle cell? They're referred to as being multinucleated. If you look at a skeletal muscle cell, you might have a nucleus here, one here, one here, one here, one here. It might be dozens of nuclei in one cardiac muscle cell. One skeletal muscle cell. Cardiac muscle cell typically just has one, kind of the normal range. The reason for that is the way skeletal muscles develop. They actually start, if you look at the development of skeletal muscles, there's actually a bunch of smaller cells early in development, and then they just fuse together and they lose all these separating bits of plasma membrane. So you had a bunch of small cells fused into one, they retained all the nuclei. Cardiac muscle cells, single nucleus. So those are just some ways to tell on a histology slide. Functionally, what else is different? All right, imagine I cut out your bicep, slapped it on the table there, what's it gonna do? Absolutely nothing. But if I were quick and I cut out your heart and set it on the table, what would it do? It would keep beating. It's a creepy thing because it does work. One of my other labs, we work with hearts out of frogs, which are very similar to mammal hearts. There are a few sub differences, but not this one. You can take a frog, remove its heart, put the heart in saline, set it on the table, that heart will keep beating by itself for hours. You take a skeletal muscle, cut it out, it doesn't beat at all. Why not? That is, what must happen for this skeletal muscle to contract? Yeah, but what makes it do that?
Voluntary.
It's a nerve impulse, right? You got a motor neuron going to your skeletal muscle. If I cut that motor neuron, what happens? You are paralyzed, right? That's what paralyzed is. If you sever the nerves going to a skeletal muscle, what does that skeletal muscle do? Absolutely nothing. It's done. We can refer to skeletal muscle as being, I think the term would be, if you want to be fancy, it's neurogenic. It means skeletal muscles receive a signal from the nervous system, from the motor neuron. When they receive that signal, they'll contract, signal stops, they relax, right? Cardiac muscle, though, that can't be the case, because you can literally go in and remove every nerve from your heart. And there are nerves in your heart, don't get me wrong, but you don't need them for contraction. If you severed every nerve going to your heart, it would still function fairly normally. As we'll see, the nerves may modify the rate, but they don't initiate it. So you can literally take a heart, put it in saline, and it'll keep beating with no connection to the rest of the animal at all. That's just downright creepy. The term for this is myogenic. It means the signal that causes contraction originates in the cardiac muscle itself. That's just weird. Other muscle cells do that, your skeletal muscles don't do that, but the cardiac muscle cells, even a single one, if you take out just a handful of cells, put them in a petri dish with the right solution, they will sit there and depolarize and beat on their own. It's the creepiest thing. I need to mention how this is possible. All right, so when you look at skeletal muscle, you went a little bit over what they call excitation, contraction, coupling maybe. The nerve impulse goes down to the skeletal muscle. There's a synapse there. It's an excitatory synapse. When it fires, it depolarizes the muscle cell. When you depolarize the muscle cell, that's what makes it contract, right? Nod your head. That makes sense? All right. So the nerve impulse, the action potential in a skeletal muscle cell is just like an action potential in an axon. It's all or none. It goes from roughly negative 70, positive something, back to negative 70, right? They're all basically the same. So in skeletal muscles or an axon, do you remember those voltage-gated sodium channels? Nod your head one way or the other. Voltage. What those were, if you imagine, here's your cell. You have a protein that we refer to as voltage-gated or activated sodium channel or gate. And remember, all those were just like a little hole in the membrane with like a lid on it. That particular protein is very sensitive to the membrane potential, right? So they had some kind of threshold. Remember that ringing a bell? If you depolarize the cell enough, the protein pops open. You now have this big hole that sodium can move through, and it diffuses in the cell, depolarizes it. That's your depolarization phase of action potential. Does that kind of ring a bell? All right, the important thing to note there is these things have a threshold. What is the threshold? That's the trigger point for this voltage-gated sodium channel. And it depends on the cell a bit, but if your resting membrane potential is negative 70 millivolts, resting membrane potential, the threshold potential might be, negative 55 millivolts. That is, if you can get the potential 15 more millivolts positive, that channel will pop open, you'll get an action potential, right? Not? Makes sense? Okay. So those have a threshold. Now we know with skeletal muscle, how do you get the threshold? Well, that's that motor neuron coming in, there's a synapse. If this thing fires, you let a little sodium in, a big synapse, and that can reach threshold, right? That's just, synaptic conduction. Same sort of thing as you get between two neurons, between a neuron and muscle cell, same thing. So to get this thing to threshold requires a motor neuron. So how does it work in a cardiac muscle cell? Because you don't need the neurons. The only difference is somehow a cardiac muscle cell has to get to threshold without the aid of one of these excitatory synapses or neuromuscular junctions. Now, the way it does that, give myself some space here. I'm going to draw myself a cell. Let's say this is a cardiac muscle cell. Now I'm going to put its resting membrane potential at negative 70 millivolts because I'm throwing new numbers. I will tell you, did you get negative 70 millivolts for resting membrane potential for axons and stuff last semester? That varies depending on the cell. Cardiac muscle cells actually have a resting membrane potential, but usually more negative, closer to negative 90, but that's not a big deal, so I'm just going to ignore it. So that's the resting membrane potential. Then they also have some voltage gated voltage-gated sodium channels, just like skeletal muscle does. There's no difference there. Second axon does. And they also have a threshold, let's go negative 60 millivolt, of negative 60 millivolts. So all this means is if I get the membrane potential from negative 70 to negative 60, these voltage-gated channels will pop open, I'll get an action potential. The only trick is we have to somehow do that without a motor neuron and a excitatory neuromuscular junction area. One more thing, something you didn't look at with the other nerves and stuff last semester. In the membrane, there's another protein. This one is called a sodium called a sodium leak channel. As the name implies, those voltage-gated channels, when they pop open, they're big honking holes, sodium pours in. A leak channel, just imagine like a little pinhole in a boat or something. It's a little tiny hole, so whatever can leak through has to go in pretty slowly. That's all we need. Because here's what will happen now. So let's say we kind of plot this. Let's have some room. We'll just put time on the x-axis, and this is voltage or membrane potential. So the blue dashed line represents the threshold. If that cell hits the threshold, it'll generate an action potential just like an axon, just like a skeletal muscle. But here's kind of what happens. All right. So we're at our resting membrane potential of negative 70 millivolts. But because of this leak channel, it's like a little pinhole, and what it does, it allows sodium ions to very slowly sort of dribble in by diffusion and following their concentration gradient. Well, as sodium moves in, what's going to happen to the membrane potential? As sodium, which is positively charged, slowly leaks into the membrane, it's going to make it start get a little bit more positive, right? So it'll slowly get more positive, more positive, more positive, more positive. Then I reach threshold. Guess what happens then? Then those big honking voltage-gated sodium channels pop open. I get a depolarization as the sodium flows in, they close, potassium gates open, and we're back where we started. And now guess what happens? Sodium keeps leaking in, sodium keeps leaking in, sodium keeps leaking in, sodium keeps leaking in. Oh, I hit threshold here again. Now I got another one. And then we're back where we started. Guess what happens now? Sodium leaks in, sodium leaks in, sodium leaks in, sodium leaks in, and I'm back to threshold. Do you get the idea? That's it. That's how it can generate its own action potentials. Your textbook, you might see these referred to as pacemaker potentials or something like that. But this is due to the leak channel. If you have these little special leak proteins in there, as the sodium leaks in, it will eventually bring it to threshold. You get an action potential, it repolarizes. Sodium leaks in, and I said slowly, but that's less than every second. You get one of these, boom, 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 because every time you depolarize a heart, it contracts. 1, 2, 3. So that's an important difference. The cardiac muscle cells are able to generate their own action potential because they have these sodium leak channels. That's why you can sever every nerve going to your heart, still be pretty much normal. There's one other thing I noticed on here. The action potentials, if you record them for cardiac muscle cells, look a little different than they do for axons or skeletal muscle cells. In fact, for you guys, a normal action potential last semester might look something like this. Right? You notice mine, I've got this big flat plateau here, and it's wider. That's actually trying to show something. The heart, its action potential is a little different. It's wider. It lasts longer. There's a couple reasons for this. I will mention the reason this has sort of this plateau at the top. There's a different kind of ion channel. It's not that big a deal, but there are what they call slow calcium gates in cardiac muscle cells. Now, calcium, like sodium, it's a positively charged ion. It's at a high concentration outside of the cell, low concentration inside. Imagine if you open a calcium gate, calcium's going to tend to flow in. Electrically, that's the same as allowing sodium to flow in, right? You're going to make it more positive. These calcium gates, they call them slow gates because after the voltage gating, sodium channels open. The sodium channels are what cause the depolarization. Then the sodium channels close, but then those slower calcium channels open and they stay open longer. So as long as those calcium channels are open, it stays depolarized. So you get this long flat plateau. Then the calcium gates close, potassium gates open, and just like other action potentials, you repolarize. So it's slightly different kind of gate, and that's important because let's look at the next thing. You remember when you're looking at the contractile mechanism, what is actually necessary to make a skeletal muscle contract? What happens? You get the nerve impulse, great, but what that literally causes the muscle to contract? You remember how it's controlled? What is the sarcoplasmic reticulum for? And what's in it in a skeletal muscle? I know it was last semester, you probably first did, that's okay. Or whatever. Do you remember the role of calcium in skeletal muscle contraction? You have to have calcium come in, bind this troponin molecule, which then move this tropomyosin out of the way. You got cross bridges and you got contraction, right? Is that ringing a bell anyway? The mechanism is the same in the heart. It has troponin, all that. Calcium has to come in, bind troponin, move it out of the way. The difference is, where does the calcium come from in skeletal muscle? Do you remember that sarcoplasmic reticulum? Nod your head one way or the other. I'll give you a quick picture up there. Well, here somewhere. This is representing a skeletal muscle. Here's the membrane peeled away. See all these blue bags and channels in here? That is the sarcoplasmic reticulum, which is basically smooth endoplasmic reticulum. They just rename it muscles. That is where the calcium is stored in skeletal muscles. So when the muscle is signaled, when it depolarizes, what really happens is calcium then moves out of the SR into the sarcomeres here, binds the troponin, and you get contraction. When the electrical signal stops, the calcium is pumped back into the sarcoplasmic reticulum, right? So it's the presence or absence of calcium in the cytoplasm that makes it contract or relax. The heart is the same way. The difference is I'm not going to write it out twice. The amount of sarcoplasmic reticulum is a little different. For skeletal muscles, all of the calcium is in the sarcoplasmic reticulum. When it's released, the muscle contracts maximally. Cardiac muscle does not have as extensive a sarcoplasmic reticulum, which means it doesn't have enough calcium stored internally. So where does the calcium come from? Remember we were looking at the hormones, parathyroid hormone, calcitonin. This is their role. Remember I said blood calcium levels are critical for heart contraction? This is what I was referring to. In the heart, while there is sarcoplasmic reticulum, it's not extensive enough. There's not enough calcium in it. Something like 20% of the calcium that's needed to come in and bind troponin to get full contraction comes from the blood, or is extracellular calcium. That's partly why I said with that parathyroid hormone, blood calcium levels are critical. If blood calcium levels drop too low, your heart doesn't have enough calcium to bind to all its troponin, it will contract more weakly. So cardiac muscle cells have less sarcoplasmic reticulum. That's one of the differences. Or, another way of wording it, they're dependent on extracellular calcium. That's also why you get part of this reason you have this delay. These are the calcium gates opening. This is actually allowing calcium to flow in from outside of the cell into the cell to bind a component so you get full contraction. All right, let's see, what else we need there? That's, that's a main event. List one. You remember what are the T tubules for in skeletal muscle contraction? Anyone remember? Transverse or T tubules in skeletal muscle? If you imagine here's a skeletal muscle cell. Remember, skeletal muscle cells are huge. When you get that action potential, this is supposed to be a synapse. That's what I can do. You get that action potential up here. Muscle cells are big. The electrical change inside the cell would even be detectable. So what you have in skeletal muscles, there are invaginations of the T tubules, which are basically just infolds of the cell membrane. What the T tubules are for in skeletal muscle is when the action potential reaches them, it's a way to conduct the action potential deep into the cell, right? So you get the signal distributed more widely. Cardiac muscle cells also have T tubules. The difference is they're bigger. So if this were T tubule skeletal muscle, well, the cardiac muscles are bigger, big hunking T tubules. Functionally, they do the same thing. It's also in the heart where action potentials can be propagated deep into the cell. That doesn't mean they have to be big though. Conduction can be tiny. This is also an avenue for the diffusion of calcium. So it's bigger, it has more surface area than a small T tubule. It's a bigger avenue for calcium diffusing from the outside. So the T tubules are a little bit bigger. All right, let's see. I think that's it for the cell. So let's move on and finish the last of the layers before I run out today. Use this one. Use this one. All right, use this one. So again, you have the parietal pericardium, its fibrous and serous layer, visceral pericardium or epicardium. Then you have the myocardium. The inner lining, what actually lines the cavities, the chambers, that's the endocardium. Go back in the diagram as well. So endocardium would actually cover the insides of the chamber, which is your left ventricle here. It also covers all the surfaces of the valves, chordae, tendae, papillary muscles. The endocardium is also very thin. It's basically a layer of epithelia in a basement membrane. That's about it, so there's not much to it. Basically, it produces a really smooth surface, so the blood moves without as much friction, creates less turbulence. That's all we need for the endocardium. All right, those are the three layers. Cell types. Now what we'll look at, more or less the same thing we looked at with the heart. Now let's go through a little quick anatomy, some of the parts. Now, as I've already mentioned, the mammalian heart is literally two pumps. I mean, you've seen it live. You can imagine if you're really good with a razor blade, you could cut through the apex here, through the interventricular septum, through the interatrial septum. You could cut the heart into a left and a right heart. There's that wall separating them, they are separate pumps. Developmentally, they happen to be in the same spot because it's easier, so revolutionarily. But mechanically, it doesn't matter if one's here, one's up here. It is two separate pumps. You could literally isolate them if you were good with a razor blade. Now, this diagram, it kind of trying to look realistic like the heart's in the lab. I'm not going to use it anymore because it kind of sucks because it's three-dimensional and it's trying to do it in 2D. You can't tell what's back here and what's up here, so I'm going to simplify my heart a bit here. Here's what we're going to do. My picture of the heart will look more like this. Want to do the left heart or the right heart first? Pick one, pick one.
Right.
Okay, right heart. So this is the right atrium, and this will be the right ventricle. Over here. Now, all we do now is kind of look at how the blood flows into the heart, what the valves are doing, where they're located, blood vessels, and so forth. So this right heart, right atrium, right ventricle, this is one of the pumps. Now, of course, if it's a pump, you can't have a pump stand alone. There has to be an inlet. So what carries blood to the right side of the heart? It would be, in a lab exam in 2 days, going into the right side of the heart would be big blue things in the cat. Yeah, which one? Superior vena cava, inferior vena cava, right? Those are inlet. Veins typically are always going to be inputs. They always take blood to the heart. So, I like it, very nice. Imagine this is your vena cava. It's coming in. There's superior and inferior. I'll draw my cat. They go into the right atrium. The veins bring blood to the heart. They actually enter the atrium, not the ventricles. So this is the inlet. The atrium is going to, the vena cava is going to bring blood into the right atrium. Now, when we're looking at the heart, you will usually see the heart's activity separated into what we call a cardiac cycle. That is, again, we already mentioned it's not a continuous pump, right? The heart ejects blood, then does nothing. It ejects blood, then does nothing. So there's a cycle to it. Contract, then relax and fill up. Contract, then relax and fill up. They have names for it. When the heart is doing nothing, it is just sitting there like a wall. That is referred to as diastole. Now, the stage of the cardiac cycle they call diastole, it's not that it's unimportant, because what you have to remember, during diastole, this is when the heart is actually filling. Filling is primarily passive. Again, you think of the heart as like a Dixie cup made out of muscle. You're just holding it under the faucet to fill it. If the cup's not doing anything, that's more or less what the heart's doing during diastole. It just sits there, passively filling. So it's literally, like a Dixie cup, you're holding it under the faucet, except it's made of muscle, and in this case, the faucet is your vena cava. So blood will actually flow passively into the atrium. And there's a big hole between the atrium and the ventricle. Remember that thing we kept sticking our fingers in, hanging over here, the left atrium, you stick your fingers in this big hole? That's the atrioventricular orifice. It's a big old opening. So blood will flow into the atria and it will passively flow into the right ventricle, just that atrioventricular orifice, that big opening there. Now, what you have to keep in mind, the atria, you've seen in the lab, how powerful are they as pumps go? They're not. They barely do squat. In fact, right now, if your atria quit working entirely, what would happen to you? Nothing. You might not even notice it. Now you might not win any marathons either, but it's not lethal. Atrias, we'll see, are minimal. They don't do much pumping. Now your ventricles, if they quit, what happens to you? You're dead immediately. But the atria are not really very powerful. And initially, when the heart is in diastole, it's all passive. The heart is just sitting there. Blood flows into the atria, fills it up, pours through this atrioventricular orifice, and the ventricle, which is your main pump, will fill to about 80% capacity completely passively. It's just sitting there. Blood just flowing in, fills it up. And we're interested in the ventricle because it is the main pump. So it will fill passively during diastole to about 80% of its capacity. Then what's going to happen? The heart will begin to contract. Well, we call this rather than using contraction, you will see the term systole. What does systole mean? Contraction. This is when the cardiac muscle cells are generating tension, squeezing out the blood, pressurizing it. And I know I already told you, all the cardiac muscle cells are connected with gap junctions, right? So they all contract at the same time? Well, I sort of lied to you. All your cardiac muscle cells do contract at the same frequency. But think about it. Does an action potential move through the heart instantaneously? No, action potentials take time, right? Well, it turns out the action potential starts in the cells of the atria, and it can't move instantaneously to every cell in the heart. Action potentials aren't like that. They're dependent on diffusion. So even though every cell in the heart is connected with these gap junctions, it takes time for the impulse to move from cell to cell to cell to cell. It takes like 0.3, 5, 0.4 seconds or so for the impulse to sweep through the entire heart. So while every cell in the heart contracts at the same frequency, if you're watching the heart contract, you'll see certain cells begin to contract before others because the speed of this impulse is not instantaneous. And it turns out if you watch heart contracting, it's the atria. The atria will begin to contract first. So when you look at diastole and systole, in general, you're talking about the whole heart. You may see it isolated in the atrial systole and diastole, ventricular systole and diastole. You may see some separation of the chambers in certain discussions. So what happens first is the atria begins to contract. And as it contracts, basically those fibers, those skeletal muscle or cardiac muscle fibers constrict, and the volume of that atria gets smaller. It's basically squeezing on the blood. Of course, as you squeeze on the blood, you'll increase the pressure. And what that does is that's going to squeeze the last bit of blood down into the ventricle. So when the atria contract, really this is what the atria do. They're hop pumps. The ventricle by itself just fills completely passively, just sitting there. Then when systole starts, it begins in the atrium. What they do is they squeeze that last 20% of the blood into the ventricles and we fill it to capacity. So as the atria are contracting, you're squeezing that last little bit of blood down into the ventricles and you fill it up to 100% of its volume. So now we are at 100% full. And shortly after the atria contract, like 0.16 seconds later, the ventricles will now begin to contract. Now you've seen the atria, you've seen the ventricles. The ventricles are massive. I mean, on those cow hearts, what are they, 1.5 inches thick? They start squeezing on blood. Now we have a potential problem here. There's a big hole between the atria and the ventricle. Now fluids, like blood, water, anything like that, they're going to move from an area of high pressure to low pressure, and they will move basically the path of least resistance. So if I have this big honking hole here and the ventricle contracts, the blood's going to try and go right back from the ventricle back into the atria. Do you want that to happen? No, because you don't want the blood to go from the atria to the ventricle, back to the atria, and back to the ventricle, and back to the, it's just oscillating. It's not moving anywhere, you keel over dead. So there are mechanisms to prevent blood from moving the wrong direction. So there's a structure between the right atrium and the right ventricle that is called, I think it was on that lab quiz up there,
The tricuspid.
tricuspid valve, right? These are the atrioventricular valves. You got one on the right heart, one on the left. The one on the right heart, this is what we call the tricuspid valve. Or you could call the right atrioventricular valve. Either one is acceptable. Now again, I think I mentioned this in lab, but the way to think of these valves are nothing special. They're just a flat connective tissue with that feeling of it sticking out there. I like to think of them as a door. You know, they're kind of hinged right here. We know they can open, or they can go the other way and close. So the way to think of valves, because they are entirely passive, you look at the door of the room. I always use the doors. But imagine this is one of the cusps of your AV valve. And you have to imagine movement of a fluid, air, water, blood, whatever. If you imagine air is blowing out of this room, what's that going to do to this door? The force of the air will keep it pushed open, right? What happens if the air changes direction and starts blowing into the room? It's likely to catch this door and slam it shut, right? That's the way the valves work. It's completely passive. It depends on basically the direction the blood is flowing or the pressure difference across that valve, which sort of means the same thing. So when the ventricles contract, the blood will try and go this direction, but it will grab the cusps of those valves and it will slam them shut. That is the first heart sound you hear. Like I mentioned in lab, blood flows through your heart in streamlines and laminar flow. It doesn't make much noise, so you can't hear it. You stick your ear in someone's chest. But when those valves slam shut, they vibrate the tissue, you can hear them. So that sound they describe as love dub or pump pump. That first one, that love, is the AV valve slammed shut. You can hear it when it vibrates the tissue. So this tricuspid valve shuts, the ventricles continue to contract, and eventually you get enough pressure, the blood has to be ejected somewhere. Well, there are two holes in every atrium. One of them, in every ventricle. One is between the atrium and the ventricle. The other hole is between the ventricle and the large artery leaving that ventricle. On the right side of the heart, what is a large artery leaving the right ventricle? It's called the pulmonary trunk, or pulmonary arteries, trunk is more accurate. But yeah, the arteries are always the outlets. They take blood away from the ventricle. So the ventricle will contract, it'll eject blood, it'll be squirting out here, but now we're gonna have a potential problem here. Do the ventricles keep contracting forever? No, they contract till they're basically close to empty. Then what do? They relax. Now, we've got to talk a little bit about blood pressure here. Blood pressure is the force moving blood through the circulatory system, but there's weird sort of aspects to it. What is a normal human blood pressure? Give it to me.
120.
Like 120 over 80. The units are millimeters of mercury. What would you do if I told you your blood pressure is 20 over 5? Are you okay?
No.
Your blood pressure is 20 over 5. Right now, sitting in this room, your blood pressure is 20 over. Here's a problem with blood pressure. Let me do my quick diagram of the circulatory system again, because your blood pressure right now is 20 over 5. There's something with this clinical measurement of blood pressure. Needs a little explanation. All right, imagine here's your heart, and here's the arteries coming out of the heart. Then you see when a cat, the aorta comes out, splits, gets smaller and smaller and smaller. I'm not doing all the branches. Eventually you lead to the capillary beds where exchange occurs in the tissues, and then it'll come back down and back to the heart, right? That's kind of my diagram of the circulatory system. Now, here's my question. If I take a pipe, this could be a blood vessel, could be a piece of PVC pipe, anything. And I'm going to have fluid run through the pipe this direction. Now, if I measure the pressure at this end of the pipe, and let's say the pressure is 100 millimeters of mercury, it's pressure measure. At this end of the pipe, the pressure is 100 millimeters of mercury. Is the blood going to flow that direction? What do you think? What makes sense? The pressure is the same here as it is here. No. Why not? Because you need a pressure difference for flow to occur, right? Fluid will flow from the higher pressure area to low pressure. So in order for fluid to be going this direction, I can tell you, I know the pressure must be higher here than it is here. If not, it wouldn't flow. Does that make sense to you? Yes? Would that make sense? You have to have pressure difference. So in the circulatory system, I know blood is moving from the aorta, right? You can literally almost watch it. It's flowing this direction, it's flowing this direction, it's flowing this direction. This is a pipe. So answer me this. If the pressure here is 120 over 80, call this place P1, and I measure downstream here a little bit, P2, is the pressure the same at P2 as it is at P1? Yes or no? If the pressure were the same, what would happen? Blood would not flow. In order for blood, any fluid, water, blood, what have you, if you're the same way, in order for them to be moving through a tube, there must be a drop in pressure. Does that kind of make sense? Which means, if I take any point going away from the heart and I measure the blood pressure, what is the pattern I'm going to find as I measure the blood pressure through this pipe? It must be dropping the whole way. Agreed? And it does. If you put a blood pressure cuff close to the aorta, measure your blood pressure. Move it further away, measure the blood pressure. Assume you have a fairly accurate pressure measuring device, the blood pressure drops through the entire circulatory system. It's highest here, it's lowest here. It's always going to be lower downstream in the tube. Does that make sense? So here's where you kind of get into issues. What do we mean our blood pressure is 120 over 80? Because honestly, it's going to be different if you move that blood pressure cuff. By convention, clinically, where do you measure the blood pressure? Brachial artery. That's 120 over 80. And honestly, in the big arteries like we looked at in lab, while there is a pressure drop, it's very small. 120 over 80, all those big arteries would be about the same. But it does drop if you have a sensitive enough measuring device. So this is the blood pressure in the brachial artery, or you can say the major systemic artery. It's not the blood pressure in the whole circulatory system. In fact, if you think about this for a minute, look at this diagram. Where is the blood pressure highest? In my ugly picture here, where's the blood pressure got to be highest? That's where it starts. Where's the blood pressure lowest? Same spot, right? When the ventricle is in systole, that is the highest pressure in the whole circuit. That's why the blood starts there. And when it's in diastole, that's the lowest pressure. That's why the blood ends up there. So you got to be a little careful with blood pressures. We are not looking at the left side of the heart or the systemic arteries. We're looking at the right ventricle. So in fact, if you could go in and measure your blood pressure in your pulmonary trunk, right now it's 20 over 5. We don't measure that clinically. It's, we'll see, it's not that important. But that is your blood pressure there. So you got to be careful with that clinical blood pressure. It depends on where you're measuring. By convention, it's a brachial artery, but pressure in pulmonary circuit is way different. I guess that was putting your books away, so you're telling me it's time for you to go home? All right. So Wednesday, that's the big lab day. For some of us, big lab and lecture day. Lucky Kayla. Studies, send me an email if you have an issue. Gail's up there. Lab's open till 5 today. Have a fun, exciting afternoon.
I've already started.
Oh, I need to stop.